So welcome everyone. My name is Doug Evans and I'm the director of the IIES, and I'm filling in today as the host because our regular host is our speaker for today. So it gives me great pleasure to introduce our speaker, Professor Cheng Gu, who is from the School of the Environment at Nanjing University in China. And he's going to be speaking today on the effects of aging on environmental behavior of microplastics. Professor Gu obtained his PhD in environmental chemistry from the University of Wisconsin-Madison in the United States. Then he worked as a postdoctoral fellow in Michigan State University, also in the United States. He joined Nanjing University as a professor in 2011, and he's currently the Changjiang Scholar Distinguished Professor and the recipient of the National Science Fund for Excellent Young Scholars. He's also the chief scientist for the National Key Lab Research and Development Plans. And his research focuses on the transformation of organic contaminants in the natural environment. He's published over 150 scientific papers in high impact journals and has been issued 15 national and international patents. I should also say that he's a board member of the IIES, and he's one of the people responsible for the formation of the IIES. So I'm very pleased to welcome him and turn the lecture over to him now. So Chen, welcome and thank you for your talk.
Is that okay for my?
You're still on mute there.
So it's okay, right? Okay. Good morning, probably good afternoon and good night. So my name is Cheng Gu. I'm from Nanjing University. I'm also the thank you, Doc, for the introduction. I'm the also in the board member of IIES. Today, I will talk about some of recent studies, some of recent studies on the microplastics. So our research group focus on the photoaging process of this microplastic. We studied the environmental behaviors of microplastic and also the subsequent ecological effects. I think every people here is consumer of microplastics. Up to now, various type of microplastics have been produced and applied in very wide field. They can be divided into some like non-degradable plastic and degradable plastics. So these are some examples for non-degradable plastics. I mean, for example, the polyethylene, polypropylene, and the polyvinyl chloride is PVC, and the polystyrene is PS. So there's several plastics. And also, right now, people use some biodegradable plastic, such as polylactic acid. This is some common plastic we use right now. So for the microplastic, usually is smaller than 5 micrometer. There are only mainly for two origin for the microplastic. One is for the primary microplastic, as specifically, which is specifically produced and used in the micro size range. They can be used for example, in personal care products. And also, most of the microplastic are from secondary microplastic. They are from the abrasion, solar irradiation, oxidation, and also some from the biological effect. During these processes, so the plastic can be smaller and smaller, becoming the microplastic. So now the microplastic problem is a serious problem. Microplastic can be detected almost everywhere in the environment. Some toxicity of microplastic, because microplastic can release toxic chemicals, chemical additives to the environment. And also microplastic, due to they have very big surface area, microplastic can serve as an absorbent for the pollutants. Then it can result drain toxicity. Also, due to the small size of the microplastics, can act as a feed food, so they can be easily ingested and transmitted through the food chain. Some common aging processes for the microplastic, such as abrasion, thermal oxidation, chemically or thermally induced oxidation, and also from the microbial adhesion colonization, also photo-oxidation process, and also some other environmental processes. I think the most important the aging of microplastic is through the photo-oxidation by the UV by the sunlight radiation. After the the aging or weathering, so the microplastic usually have the color change. Also, the surface is become rougher, and also the hydrophilicity usually increase. So the question is that when the microplastic undergo aging process in different metrics, so whether there are some differences. So we will start it. This is our objectives for we want to start it the photo aging processes of some typical microplastics in water and also in the air. So the first study is we started the photo aging of microplastic in water. So in this study, we use the PVC as an example, because the PVC is widely used, and it can they can be used as film, pipes, and also used as different bottles. So in China, China is becoming the #1 PVC producer in the world. So studies some organic low molecular weight organic acid, such as oxalic acid, citric acid. So when they present in the environment, so they can when they irradiate by the light, especially for UV light, they can generate the carbon dioxide radical. Because because the carbon dioxide is very this radical is very reactive, they can then react with oxygen to generate the oxygen oxygen hydroxyl oxygen ion, the radicals, and also becoming the hydroxy radical. So for both of them have high potential to can accelerate the accelerate the aging process for the microplastic. This from the previous study. So this is our the schematic diagram for our experimental setup. So in this process, the UV PVC microplastic suspension. So in the presence of this low molecular weight organic acid, and also also in the presence of iron 3. This is because when they form this complex from the previous literature, they can generate more reactive oxygen species. So this we want to study, the PVC microplastic, the photoaging process in the presence of this low molecular weight organic acid, and also in the presence, in addition of iron 3 cation. So this, during the photoaging process of this PVC microplastic, if they release the chloride, it indicates the structure, the surface structure will change and also indicate the agent start. So then we look at our result, we can see that. So we use for example, use oxalate and also use citrate. This in the presence of this oxalate and iron, it has highest the degradation for this PVC microplastic, as indicated by the release of chloride. So also for the for citrate also have the similar we get a similar result. However, with only the iron and also with only oxalate, so but with only iron, so the enhancement is not obvious, but with some oxalic acid, so they may have some enhancement, but the most significant increase is in the presence of oxalate and iron 3. Then we studied different radicals in the system. We use EPR spectra and also use NBT. This is a common reagent to detect different radicals. So we found that in the system we have the carbon dioxide radical, hydroxy radical, and also has the oxygen iron radical, anion radical. Then we did some quenching experiment. We found that the hydroxy radical is play the most important role for the PVC microplastic degradation in the presence of this low molecular the oxalic acid and also oxalic acid with iron 3. Then we also did some FTI analysis for the system. So during the photo aging, as with different time, with longer reaction time, you can see more carbon double bond, and the hydroxyl group and the carbonyl group appeared after photo aging. And also we can see from the FTIR spectra, in the presence of oxalate and also iron 3, it can significantly accelerate the generation of unsaturated alkene, the carbon double bond. Then we propose so the different aging pathways. So this the first two is first one is by direct UV light radiation, the dechlorination reactions. So the PVC under the UV radiation, they can release the chloride and also form the radical. Then they can form radical and continue to lose can lose proton and also chloride. They form the carbon-carbon double bond. And in the presence of oxygen, so they can in the presence of oxygen can be this oxygen molecule can be involved into this the aging process. So they'll form the carbonyl group and also hydroxyl groups. However, with the presence of this oxalic acid and iron, they can generate hydroxy radicals. So they can follow different pathways. So they form more this carbonyl groups, and also form some different with more hydroxyl groups in the structures. So this for the conclusion for this part is that this hydroxy radical generated from photolysis of low molecular weight organic acid and also their ferric complexes could accelerate the transformation of PVC microplastics. Then we talk about, we started the photoaging of microplastic in the air. Because in China, like for agriculture, we use lots of plastics for to cover the surface, the surface. So, but this surface is not water saturated. So we all started, like in the photoaging processes of microplastic in the air. So without water. So the humidity of this aging system was controlled. So we want to see the air humidity, whether the air humidity can affect this photoaging process. To study this process, we developed a system. Like in this system, the humidity, relative humidity of system can be accurately controlled. By controlling the gas flow in different channels. And like the microplastic particle was fixed in this place. This so for this environmental setup, we can observe the changes of this microplastic during the photoaging process in situ. The surface change in situ by this like by this specific system with the FTIR system. So in this system, the relative humidity of the system we can control between 10% to 90%. So the result shows that the carbon-carbon double bond, carbonyl and hydroxy groups are formed on the surface of this PVC and also the PS microplastic after the photoaging. So and also very interesting phenomenon is that the contents and the generation rates of this carbonyl and hydroxy groups significantly varied as the pH as a relative humidity change. As relative humidity increase from 10 to 90%, the production of carbonyl decreased. On the other hand, the hydroxyl group increased. So in addition, the relative humidity exhibit a greater effect on PVC than the PS. Then we also did some XPS analysis, also confirm this relative humidity effect on the formation of oxygen containing moieties. So as mentioned above, relative humidity shows a very significant effect on photoaging of PVC microplastic than the PS microplastic. So as shown in this figure, the PVC microplastic surface is more hydrophilic than the PS. A hydrophilic surface could provide more absorption and reaction site for water molecule. As a result, PVC surfaces are more fissile to be influenced by water molecules. So this is our explanation. So then we proposed the reaction pathways of PVC and also PS microplastic and different relative humidity conditions. At the low humidity condition, water molecules do not participate in the reaction. The PVC and the PS undergo a certain aging pathway to generate carbonyl and hydroxyl group. However, at high relative humidity condition, so water molecules are involved in the reaction pathway, leading to the branching in the reaction pathway. reaction pathways with water molecule involved are more feasible for the generation of hydroxyl group and suppress the generation of the carbonyl groups. Because the relative humidity could significantly influence the typical microplastic photoaging processes and subsequent physical chemical properties. The environmental behavior and the effect of aging microplastic should be evaluated. In these studies, we investigated the absorption of commonly used antibiotics. In this case, is tetracycline on the aged microplastic at different relative humidities, as shown in the figure. So the absorption of tetracycline on PVC is significantly affected by the relative humidity. However, the relative humidity showed negligible effect on the absorption of tetracycline on the PS microplastic. So the negligible effect on the absorption on PS could be attributed to the negligible changes in the PS surface hydrophilicity, as mentioned above. So the significant decrease in the tetracycline absorption on PVC microplastic is proposed to attribute it to the different bonding modes between tetracycline and the PVC microplastic surface. So we also use in situ FTIR to explore the surface interaction. So the PVC aged at high relative humidity can form more hydrogen bonding. Could form at low PVC aged at low relative humidity could form a hydrogen bonding with the carbonyl group in the C ring of tetracycline. However, the PVC aged at higher relative humidity can form double hydrogen bonding with the amide group on the tetracycline. Because the double hydrogen bonding is stronger, so the PVC aged at high relative is more feasible to absorb more tetracycline. So this is also consistent with our experimental result. So this is give a conclusion. With the increase of this humidity, water molecule can participate in the photoaging photoreaction process of microplastics. Thus, the absorption effect of tetracycline on the PVC microplastic at different humidity will be different. So this also due to the like at high relative humidity, more hydrogen bonding more hydroxyl group are formed on the surface, so they can provide more hydrogen bonding. So that's the reason why more tetracycline can be absorbed. So the next is we studied the effect of microplastic and the degradation of some pollutants. So in this case, we choose widely used antibiotics is cephalosporin. So this the degradation of this antibiotics has been well studied. So they can the amide bond here is broken, can be hydrolyzed, so to form this kind of structure in B. This is a hydrolyzed the molecule, the hydrolyzed structure for this hydrolysis product. So this is it's just for the amide group. So then initially, so we photo aged the PVC microplastic. As you can see here, after photo aging, the surface is becoming rougher, and also surface is more hydrophobic, and also has a more carbon-carbon double bond, carbonyl groups, more carbonyl groups, and more hydroxyl group on the edge the microplastics. So also by the like the XPS analysis also same. So very interesting result shows here. So when the PVC microplastic was added into the system, so we can see more hydrolyzed product are formed. So like so with a different so here if you look at here, so the for the control experiment is no plastic. There are certain hydrolysis reactions, and also in the presence of the pristine PVC microplastic is no significant increase for the hydrolysis reaction, but with photo-aged PVC microplastic, you can see significant increase for the production of hydrolyzed product. Then also compare the like the FTIR spectrum for the pristine PVC microplastic and the PVC aged PVC microplastic. So the when they interact with this the CFZ the antibiotics, so we can see that most CFZ can be absorbed on the edge of the PVC microplastic than the pristine microplastic. So we also can see the hydrogen bonding of this carbonyl group in the beta lactam and also the carboxylic group on the CFZ to the surface hydroxyl and the carbonyl on the surface of aged microplastic are formed. It's clearly we can see the peak shift, and also the peak is becoming wider. So it indicates that they can form some kind of the interactions, mainly for the hydrogen bonding. We also do some theoretic calculation. So we simulate the intermolecular interaction and also the geometric structure of this CFZ antibiotics with PVC microplastic. So the positive molecular charge of the carbonyl carbon atom in the beta lactam ring was increased from 0.563 to 0.579. The significant increase of this molecular charge indicate that the hydrogen bonding forces between the CFZ and the aged PVC microplastic can facilitate the water-induced nucleophilic attack. So it will induce the hydrolysis reaction. So we also did some theoretic calculation. We can see that. So the hydrolysis reaction activation energy of this CFZ when they coexisted with PVC, aged PVC, and also with in the presence of water molecule, was significantly decreased. So this also can provide some convincing evidence for the enhanced hydrolysis of CFZ by the aged PVC microplastic. So we do give a quick conclusion in that the photo-aged PVC microplastic can promote the nucleophilic attack of water molecule by forming the intermolecular hydrogen bonding with the CFZ antibiotic, and also can accelerate the hydrolysis reaction. So next study is the fluorescence and also biodegradability of aged microplastics. So in the current knowledge of environmental behavior and toxicity of microplastic and nanoplastic is limited by the external aid of fluorescent staining analysis technique. Because when you use this technique, sometimes like the fluorescence stain can be be lost during the analysis, so they may have some wrong result, provide some wrong result. So they can when you use the fluorescent dyes, so the disadvantage is like they can sometimes get a false positive, and also high cost, low efficiency, and some other disadvantages. So we want to see during the photolysis of this microplastic whether they can form some kind of the fluorescence, and also probably increase biodegradation of this aged microplastic. In these studies, we use four common plastics, microplastics: the PE, PP, and the PVC, and PS. And we use Daphne as the model, like animals, test animals, because this animal is easy to be easy to be raised, and also they can grow very fast, and also cheap. And also this Daphne is very sensitive to some external changes. It's very commonly used to evaluate the aquatic toxicity to study the ecotoxicity. And also it has a transparent shell that can easy to be observed by the microscope. Then we studied the four microplastics. We can see that and this is under different radiation conditions, such as under natural and also under solar radiation. We can see here, in our system, we did some UV radiation. So after like 72 hours radiation, so the full microplastic all develop the fluorescence, especially for the PS and the PVC microplastic. So this top is under simulated radiation conditions. On the bottom is under natural solar irradiation. So we can see both in both cases. So after a certain time radiation, UV radiation, they can develop some fluorescence on the microplastic surface. And also see the very similar results. So after aging, the surface become rougher, and also the contact is more hydrophobic in this after photo aging. We also did some FTIR spectra, shows that in the four microplastics, so more we can see the carbon-carbon double bond are generated after photo aging. So this might be the fluorescent unit, because with the carbon-carbon double bond, they can have the pi the bond, so electron can be easily accelerate, can be easily excited, so to form the fluorescence. That's the probable reason, because they form the conjugated carbon-carbon double bond. on the surface of this microplastic after photoaging. We also, we use the fluorescence microscope. We can see clearly on the for the surface of this PS and the PVC microplastic after for certain hours photoaging. We can see clearly the fluorescence. Then we do the microplastic exposure on the Daphne. This is definitely born less than 24 hours after culture. And the enrichment of the intestinal microplastic can be merely observed. So when you look at here, is that so this is original the Daphne, when the starving Daphne, so it's no fluorescence, and also when they exposed to the primary microplastic, it's also no the fluorescence. However, when we expose to the aged microplastic, we can clearly see the when they expose to the aged PVC microplastic or nanoplastic, we can see clearly in the intestine, we can see the fluorescence. So we also did some dynamic processes of ingestion, and also did the 84 hours excretion of this aged PVC microplastic. So after 24-hour ingestion, PVC microplastic filled the whole intestine, while after like a 48-hour excretion, there's still trace amount of PVC microplastic remain in the intestine. And also compared to the microplastic PVC microplastic, for nanoscale microplastic, especially in this in our study, we use 100 micrometer, like the microplastic. So this size, the smaller size, even can cross the Daphne gut and enrich, in the body's tissues and some organs. So it's also it indicates that with smaller size, especially for the nano-sized microplastic, it may have higher toxicity. And also very interestingly is that when we when the aged photo-aged microplastic is fitted to the Daphne, so the carbon signal decreased. At the same time, the carbonyl and the hydroxyl groups are enhanced. So after ingested by the Daphne, this photo-aged microplastic becoming rougher, and also the oxygen carbon values also increased. So the carbon the oxygen content increased and the chlorine content decreased. It indicates that the aged PVC microplastic probably is more biodegradable for the Daphne. So when you look, however, when you compare to the pristine nanoplastic, so after intake by the Daphne, its surface is unchanged. That's very interesting. So we consider that after photoaging, so the surface of this, like the microplastic or nanoplastic, is becoming more hydrophilic. They probably can be more easily to be uptake by the Daphne and get even more, even more aged in the body of this Daphne. So I think this is very interesting phenomena, and I think we are still starting for further studies. So this is also our recent publication. So thank you very much for your attention. So now I'm ready for the questions, Doc.
Okay, thank you very much, Chen, for that excellent talk. So yes, we're open for questions. I'm sure Chen's willing to answer any questions that people have. Maybe while people are thinking about it, I'll ask you a question. And this is kind of a big picture question.
Okay.
So in one sense, photodegradation of plastics could be seen as a good thing, because we're breaking down some of the structure. But a couple of weeks ago, there was a paper published that looked at rotifers and how they interact with microplastics. And one of the conclusions of that paper was that a single rotifer can produce up to 35,000 nanoplastic particles per day. And one of the things they found was that photodegraded plastics were more easily broken down into nanoplastics by rotifers. And so the question becomes, are we better to try to make plastics that are photodegradable more rapidly, or in some senses, if we do that, are we actually making it more harmful, and we should actually be just looking for ways to remove the larger plastics out of the system before they start to photodegrade?
Yeah, thank you, Doc. I think that's very excellent question, because actually in our group, we mainly studied like natural processes, and it's true, after photoaging, the plastic is will be more fragile to be to be broken into like even smaller part, for example, into the nano particles. They may induce like a higher toxicities to the ecosystem. So that's probably true. Like yeah, I agree. Like the problem we need to avoid, like the negative effect of photoaging. We probably should like collect the plastic to do not want them to be degraded into smaller pieces. And also probably what we can do is that we can use some biodegradable microplastic, because they may be easily degraded and to mineralize. So that's the ideal. But in some cases, for the biodegradable microplastic, even more toxic, because it's easy to be degraded into smaller pieces. So it's it's very difficult question, and I think more studies should pay more attention on this natural processes. Doc.
Cheng, hey, this is Aaron. How are you doing?
Good, Aaron. Long time no see.
A long time no see. Great talk today. Couple of questions. One is, I just came back from CTAC in Louisville, and Doug, this is a question for you as well because it involves metals. But there, a bunch of researchers at Duke are doing some interesting work with sea anemones and PVC, and they're exposing pellets to sea anemones, and they're actually getting in the body of the sea anemone detectable limits of above detection, so detectable concentrations of tin. So that's kind of concerning, I would think, that these anemones can actually extract tin, because we know tin's an endocrine disruptor, et cetera. As they degrade, do you think that this is happening in the natural environment, that tin is actually leaching from the plastic? Because I don't know the metal, Doug, how the binding would happen. I guess they use organotin and lead in the production of PVC.
Yeah, there are like areas. So I think like it's very also interesting, because we didn't study metals, but we did study the the plastic additives, such as the PAE. So this kind of this kind of organic compound, they can be released, during the aging process, because this, like for the PVC, for example, for the PVC, it's up to 30% is additives. So maybe they also have some metals too. So during the aging, so they are more surface exposed. So the metals and also the additives can be released. I think that's probably, yeah.
Okay.
So we also do see the in our studies, we do see the they're releasing this type of chemicals.
Okay.
Yeah, I was just going to follow up on that also, Aaron. If I'm not mistaken, I think that they use quite a bit of tin in the curing process for PVCs, and so I guess the question becomes: is that firmly attached to the smaller particles as they're broken down, or is in some way being released and being just uptaken as a soluble form? And obviously, I don't think we know that at the moment.
Yeah, I just thought it was interesting that these little pellets were the anemone was extracting enough tin to be detected in the whole body, which was quite amazing. Thank you. Just the last question. As you know, I'm reviewing a lot of papers on microplastics, and I do see a lot of papers with accumulation and of microplastics in various organisms, such as Daphnia magna. And I guess the question is, this is, I guess, big picture question, like Doug was looking at. When do you think we reach the tipping point where organisms like microorganisms like Daphnia, et cetera, are starting to accumulate enough to cause effects? Because right now we know that zooplankton is still far more densely, concentrated than microplastics. Do we have 10 years, 20 years, 100 years? What do you think of that?
Actually, like in our study, we do see that when you do the when you expose exposed to the microplastic, because they can use it as a food, I think, so they can accumulate in the body, right? So they but like after feeding, when you do the they do the excretion process, so it can be excreted from the body. But if you have the continuous exposure, they may accumulate more and more, I think. So they may form, but the basically, I think the smaller microplastic, like the nano microplastic, may be more toxic, because as we can see that, so the small nanoplastic can penetrate the gut, to the tissues, and also probably to some organs. They may form some more toxic effect. So also depend on the size too. So
yeah, I guess maybe the question was a bit confusing, but I guess the problem is a lot of the papers that we're rejecting, they're overloading these organisms with microplastics. Yeah. So they're really setting up not realistic levels of exposure. So, and of course the organism is not living because it's it's loaded with plastics and it can't excrete it. So anyway, okay. Thank you. Yeah. Great talk today. Thank you.
Okay, thank you. Thank you. I think that's the problem, that the people want to see the effect most significantly or easily or quickly. That probably.
Do we have any other questions for Chen? All is clear then, I guess.
Okay.
Well, if there are no more questions for Chen, let me once again thank you for that very interesting talk. Really appreciate it. And just to let you know that our talk next week, Claudia Halsband, who spoke earlier in the series, will be back, and she's going to talk about some very recent work on tire wear and microplastic particles coming off tires as they wear. So I hope that you'll be able to join us then as well. So thank you again for joining us today, and we'll see you all next week.
Okay, bye-bye.
Bye all.